to meet you. Hey, Brian. What are you doing here? Just hanging out? All right, cool. Good morning, everyone. Can you hear me? More or less through my mask. Hey, all right, so let's get going. See if my high-tech pointer here works. It does. Hey, so on Friday, so today we're going to finish up the elazobranchs, looking at the squalid elazobranchs, which are the, I should put this down if I'm not going to use it. The squalids, which are the skates and rays. And then we'll go on to the actinopterygii, which are the ray-finned fish, some of the early primitive ray-finned fish, and look at the evolutionary advantages as they emerged. And then hopefully, if I move along quickly enough, which is unlikely, we'll get to the teleosts, which are sort of the hey, will you grab the door for me when you come in? Thanks. The teleosts, which are sort of the new updated model of fishes in the evolutionary scope of things. So that's our plan for the lectures. Draft of the paper is due tonight. Thanks for getting the door. Draft of the paper is due tonight. And so you're gonna submit it. You gotta submit it. I don't care what you got, just turn it in. If it's really crummy, you'll get really mean reviews. If it's really good, you'll get really mean reviews. Not really. No, I'm going to talk about that. But you got to submit it. It's 10 points. And you can't really move on in the game. It's kind of like Monopoly. You got to get out of jail. You got to spend your time. You got to submit the draft because you can't progress to the next thing. Unless you submit a draft, and I'm not going to take late drafts because you're going to put it into the beast that is Canvas, and it's going to get rerouted to each of you. You each should receive 2 drafts that you then have to peer review. You got a week to peer review it, so by next Wednesday. So you've got to read both of them, and you have to give some feedback. And here's how you give peer review. It's a cordial process. It's not brutal. And you want it to be positive feedback, unless it's really crummy and then maybe a little bit of shade. But mostly you want it to be positive feedback, and here's how you do it. You read the draft, and then you take some notes and you identify issues by line number. And the first paragraph goes, it was my pleasure to read this paper by such and such an author with such and such a title. And then you add a couple of nice things in that paragraph. Doesn't matter how bad it is. You just say a couple of nice things. It might be a stretch. And then the next paragraph is going to be a summary of what the paper was about. So the key details, obviously it's about a fish. It's not a big stretch there, but you want to name the fish and some of the points. You want to look at that list of things that we asked you to submit, and you're going to say, well, the authors talked about the morphology, the distribution, the behavior, bingo. You know, just shows that you read it. It's standard operating procedure with a peer review. Show that you've read it. Be nice, first paragraph. Show that you've read it, second paragraph. Third paragraph, you go through and state the overall issues with the paper, the big picture issues, like organization. Could they have organized it better? Don't say, this paper was organized like a piece of trash, because it's not helpful. But you know, people do mean reviews. I got some, I've seen some. Don't do that, please don't do that. Instead, say, here's how you could organize it better, and give them points. It doesn't have to be a long paragraph, it can be bullet pointed, but just clearly identifying how they might organize it better. Or if their language was bad, suggest that they get somebody else to peer review it, somebody else to proofread. If there's a lot of grammar issues, point out that they could get a proofreader before they submit it. And then the fourth paragraph are the microscopic line-by-line items that you found that you could call out improvements on. And for this, if you're really gracious, you might include some glaring grammatical mistakes. It's not really on you to catch every period and comma and quotation mark, but you know, you might want to point out some of the glaring grammatical mistakes if there's a manageable number of them. And if there's any kind of like ideas or something that weren't clear, a sentence that was unclear, Just call it out and state it very plainly, very neutral. The thing is to be positive, to be honest, and to be neutral, because people don't receive criticism well when you say, really cutting things. And it's just sometimes when you read a paper, it's just so infuriating. You're like, why did you do that? And so there's this huge temptation to troll the person. To resist it, just like hate mail, like emails are like that too. Such temptation. I'm just gonna Facebook. I'm just gonna post. Don't do that. Be nice. And then once you submit it, Wednesday, you got to submit it. You got to submit it on time. There's no late stuff. Each section's worth 10 points. Submitting it today is worth 10 points. Your peer reviews are going to be 10 points for your peer reviews, both of them. And then you get the paper back, and each of you will be able to review the comments. And then you'll have, like, I think, a week and a half, 2 weeks, something like that, to submit the final paper. That's the process. Any questions? Yes. Does the what have to be the references? The closer you get, the better off you are and the easier it is on your reviewer. So do your best, but you still have time to modify. Mostly you're asking for feedback on the structure of the paper, right? But drafts or the citations, the fair game. If you find a draft paper and the citations are all botched up, then it's on you to call it out, right? So you might get feedback on that. Yes. Yeah.
That's like kind of general, like this is the largest fish in the genus. Should we cite like every
you can
text, or should we just pick one?
You can, sometimes people cite the earliest paper that states that. If it's a more complicated idea, or a more nuanced idea, and people have built on it, like the jaw structure in this fish, and how the autostylistic jaw works, that might be a more recent draft, even though people have suggested things before. Might be a more recent, updated version, so you might cite that, or you might cite all of them. It's okay. Other questions? Yeah.
Are your reviewers going to grade your paper?
No, it's not going to be graded. It's just for your feedback so that you could write a better paper. There's no grades on the draft other than submitting, and there's no grades on the peer reviews other than submitting. And the peer reviewers have nothing to do with the final grade on the paper, which is a separate grade. So 3 grades in total, okay? All right, I think that's it. There's an article on Bashir's that would be good to read tonight. So before we come back on fish skull structure, which I'm gonna talk a little bit about today, and this might help clarify some of the things I talk about. And then another chapter in Helfman et al., chapter 14, teleos, and then I'll have a quiz on Friday, okay? So I want to go back to one of the old participation questions. For each group, offer 3 reasons that may explain why they went extinct. So number 1, the ostracoderms, the early jawless fish. Why might the ostracoderms have gone extinct? What liabilities did they have? Presumably you filled out the participation questions, and so you've thought about this a little bit. What is an ostracoderm? Hello? Yes. Yes, it's a fish with a bony armor. Yeah, that's true. Yes. Yeah, there's a common thread here. Yeah, they're all pretty heavily armored, and that would be the same thing for each one. Each one of these has, this one has heavy bony plates or scutes on the exterior. This one had a really, this is a placoderm, had this very armored head, these giant teeth. It was a giant fish. And the acanthodians, which are their own sort of shark-like, sort of fish-like group, they have these weird, heavy-duty spiky projections. And what at one point was a defensive mechanism became not that useful anymore. Why? What happened in the world that may have made bony, heavy plates less useful as a defensive mechanism? What's the liability? Yeah. Yeah, so this is the agnathan. So the lamprey and the hagfish, they were light bodied. They didn't have the armored plates, and yet they ended up being more successful, possibly because they were lighter, easier to find refuge, easier to hide. It turned out that having the plates was not such an advantage. Okay, so fins that weren't like not functional for mobility or something, so made them less able to compete with other organisms. Yeah. That's right. That's right. So these big kind of grasping jaws became pretty obsolete and outdated. Anything else? Yeah.
For the plesioderm, is it possible that the teeth are kind of used to so that, like, for example, with the great whites, like they can lose their teeth over time and they'll come back, but these ones probably get worn down.
That's a very good thought. I don't know. That could be that they didn't have replaceable jaws or replaceable teeth because it was built into the jaw and they were so energetically expensive to produce giant teeth like that. That's speculative, but this is the game of speculation. We're talking about ancestral fishes from the fossil record. I could say also this placoderm. This occurred in the late Permian. There was an atmospheric disruption, which presumably corresponded with the demise of this particular group. And that in general is another thing that happened to all three of these. Climate change, a changing planet, changing opportunities, as well as the evolution of competing fishes. That is the teleostean fishes, fast, lightweight, ideal predators. Any other thoughts about this? Yes.
What about like the extinction of like some of the fishes and then maybe your bony armor or is that?
Oh, interesting. So like after the Permian extinction or even the KT extinction, you no longer have this sort of intense predation by giant predators. Yeah, that could very well be that there was a shift in predation, that the shift, it's all kind of like a lottery, right? You kind of roll the dice of evolution and see what comes out of it. And, increasingly over time, evolution seems to have favored faster, lighter, and smaller, really. And as we've seen that on terrestrial animals as well as the aquatic, that lightweight, fast predators are very effective. Another thing, yeah?
Could it have also been like the teleostean fishes could reproduce much faster and produce way more offspring? So these guys, maybe produce 1 or 2 offspring and could compete.
That's an excellent point. We find that, we don't know much about the reproductive capacity, but we do know that in sharks, they have mostly ovoviviparity, which is live birth, right? And so in some senses, that might be a liability. I'll probably get to that today or tomorrow or Friday. Yeah. Yeah, that's right. I think so. Energetically expensive to make them and energetically expensive to keep them and move around with them. Even though you had massive, predation resistance, it may just not have been effective when competing. And then there may be lots of things we don't know. These are the kinds of broad, open-ended questions I will ask on the exams, right? So they get you to actually think about evolution, thinking deeply about how these things, the idea is not to like memorize everything about a fish. The idea is to be able to think about evolution and conservation and the environment and the ecology and how the species that you're looking at interact in a synergistic way. So hopefully we'll get there eventually, okay? I'm gonna move on now. And that's what these sorts of questions are. The participation questions are opportunities for you to kind of think and collaborate and talk to people about it. Okay, so my last lecture was kind of a dud. It was like I just threw a whole bunch of information at you and it felt like it felt very random. It was like fish trivia night. So I just want to tell you that when I'm talking about the orders, so we're going to be in the lecture, you need to know down to the order. In lab, you need to know down to family. And I'm going to basically arrange the slides, that is the taxonomy slides talking about each order, kind of like this. So I'll have listed the families, the number of families, genera, and species, or at least families and species. I'll include common names. and then key defining characteristics. And key defining characteristics, and then some evolutionary information. And then in a box, I'll have key character. So that's about the order. This is about just the general order of the fish. And then for the families, I'll have discussion of physical morphology, size range, feeding, and ecology. And sorry, my pointer just isn't really that high tech. There we go. And then I'll give you a specific species example with the distribution in a little map or in words, and then its status here for the IUCN status. And then I'll usually have a picture here too. And this picture actually is our art thing for today. This is Pablo Picasso, lemon, fish, eggplant. You know, Picasso dealt with you know, he pulled back from the extravagance of impressionistic painting and became an essentialist. He tried to capture images for their essential piece that made them what they were. And later, as his art progressed, he tried to capture not only physical essential items, but also emotional essentialism to capture and distill a feeling, which became the foundation for a lot of modern art. And in this particular picture, the fish is pretty interesting because we know it's not just a doodle of a fish entirely. It's got two eyes on one side, which means it's a flatfish of some sort. And not only is it a flatfish, we can tell with the way the mouth goes that it's a left-eyed flatfish. And there is a Mediterranean left-eyed flatfish. Picasso was from Spain, spent a lot of time in France. It probably was a wide-eyed flounder, which is found in the Mediterranean and one of the few left-sided flatfish. You can tell it's on the left side by the shape of the mouth. And so with a few lines, he sort of distilled it into its essential pieces. It's, you know, what it is. What's important about it? Well, it's shaped like a fish. It's got eyes on one side, and you can identify the species as a result of it. And that became, that was a reference back to ancestral art, cave painting, a lot of African masks and stuff are all things that he referenced in his art. And that's fish art for the day. So moving along. So today we're going to talk about the squalids. We got through the galiad sharks in the elasmobranchs, and now we're going to do the squalids, which are basically shark-like or ray-like fishes that have cartilaginous skeletons. And these are the five main orders that I want you to know: the squaliforms, or dogfish sharks, the rhinopristiforms, the sawfishes, and the this thing is not working very well for me. I'm trying to do this so that people at home can follow when I point. But the rhinopristiforms, the sawfishes, the torpediniforms, the electric rays, the rajiforms, the skates, and the myliobatiforms, the rays. And so I'm going to swing through these basic groups. Squaliformes includes the dogfish, sleeper sharks, and cookie cutter sharks. So they're shark-like rays. They're more shark-like than ray-like in this particular group, and they're sort of a cusp species between the galiad sharks and the squaliad sharks. They're basically, they have two dorsal fins with spines, which makes them kind of a unique group. No anal fin and no nictitating membrane. That's the membrane that cats have that cover their eyes to protect them from prey. These sharks lack a similar membrane. And they have 5 to 7 gill slits. That's sort of identifying for this order. And a typical family for this group, there are 7 families, but a typical family are the dogfish sharks, the squalids. These guys have venomous spines. They tend to be pretty small, and they're piscivorous. They hunt in packs. At least in the early accounts of these, people said they were like dog packs, hunting collectively up on the shallows. And so they got the name dogfish or dogfish sharks. They're migratory species, and they're ovoviviparous. They gestate up to 2 years before they give live birth. And a classic example of this here in the Pacific, especially on our coast as well as Asia, is the Pacific spiny dogfish. And it's a low-value fish, so low-value fishery, so it's got a stable population. You can see that it's, if you can see here, it's a species of least concern. Okay, The next order is the rhinopristiformes. These are the guitarfish, shovel-nosed sharks, banjo rays, and these are more like ray-like animals. And as we go down these groups, you'll see they become more and more ray-like, that is, depressed dorsally to ventrally with wider and wider pectoral fins that attach to the head. And so the characteristics for this group, like all the other rays, include mouth, nostril, and gills being ventral on the other side. They have extremely pointed rostrum with the sawtooth that you see here. And the pectoral rays are fused to the head. These two are oviviparous, which are many of the sharks and rays are oviviparous. This is example here is the large tooth sawfish, Pristis pristis. It's a representative of the family Pristidae. It's probably the largest member. Presumably, this species can get up to 7 meters. And I would say that because of its endangered status, it's critically endangered. One rarely sees a fish like this. They tend to be in decline everywhere. They're overfished for their fins, for the saw, which is sort of a totemic item in many cultures historically, and for meat. And I can tell you that the overfishing probably is not as a result of regional groups fishing for cultural practices, but rather the demand for meat has pushed them towards extinction. Torpediniforms are the electric rays. There's 4 families, 69 species. There's a lot of electric rays out there in the world. They tend to be of least concern in terms of their threatened status across the board. That's true of arrays because everybody's a little bit afraid of them. I'm not a big fishery for them, possibly. The characteristics of these guys, in addition to having these electric organs, which are a muscular organ, they contract the muscles and then release an electric charge, which can be quite shocking. I'm sorry. They have a rounded pectoral disc, so they're very, very round. They have this very muscular caudal fin, so they're a ray with this very shark-like posterior, essentially. The body is very flabby. It's totally notable when you see them. They're just kind of chunky. And then, like all rays, they have the mouth nostrils and mouth ventral with spiracles on the dorsal side. Family Torpinidae is one of the 4 families that's represented here on the west coast by the Pacific electric ray. And they can grow to be pretty big size. They don't have a very good vision, and the reason is probably because they live in very turbid waters and they have an electric field. So the electric organ is not just for shocking prey, but it's actually for sort of visualizing the landscape around them, as it were. Yes?
The word oviviparous, is it the same as
No. A viviparous is like a true placental arrangement. Oviviparous means that they have an egg that they're gestating inside. And that egg, after it depletes the yolk, that egg-like structure often gets attached to a pseudoplacenta, but that's the difference. Oviviparous, viviparous, and oviparous. Very confusing. And so, yeah, they give live birth. Oh, and back in the 1st century, it was recommended famously that they could be used for headache pain, because if you hurt yourself hard enough, your headache is going to go away. And also for hemorrhoids. And I just can't even, I don't know. Have you tried that? No. Okay. I haven't either. I don't recommend it, but they do hurt, like an electric fence. Okay, so order Rajiformes are the skates. They have a depressed body, so they're much more extremely depressed. They have characteristic of this group is that they have these very the pectoral fins are even more fused. They kind of have a triangular type shaped head and pectoral fin structure because the pointiness of the rays, as opposed to the roundedness of the electric rays. And again, like all the rays, everything's ventral, right, except for the spiracles in the eyes. And they have a characteristic motion called rajiform locomotion, which is this kind of cool undulating. It's a very kind of very delicate undulating of the rays, the pectoral rays, as they cruise along the bottom, searching for food. And they tend to be benthic feeders and plankton feeders. And so that helps them move along the bottom using a minimum amount of energy. And so this is a really huge, these skates are a huge order and also a huge family, I should say. They make up 20% of all Contrichthyan diversity. They sometimes have spines on the dorsal side. These are oviparous and they lay these, what we call mermaid's purses, which you may have collected on the beach. And each one has an embryo. Usually it's just one embryo inside. Sometimes it's two. And they attach these to structures on the bottom so that they can hatch independently. And they also have an electric organ. It tends not to be as strong as in the electric rays, but they do have one, and it's mostly sensory. Big skate is our local one, and it's also not a terrible concern. The Mylobatiformes are the bat rays, the eagle rays, the sting rays, and the manta rays. So these are kind of the impressive kings of the ray family. And this is where, with these rays, you can see the sort of ultimate flattening, this really depressed form with very extravagant wing-like rays that they use to kind of fly through the water. There's both benthic and pelagic forms, and they often have a venomous spine. It's also serrated, as if the venom's not enough. It's a venomous serrated spine, not all of them. But when you step on them, sometimes they'll stab you with that. You ever been stabbed by that? No, neither have I. I don't know what it feels like. We'll have to try it out sometime. It can really hurt. These animals tend to be oviviparous. They have very low fecundity. There's both marine and freshwater species, which is somewhat unusual. And they're increasingly threatened. These species tend to be, as a group, overfished. And on top of the overfishing, there are sometimes in some countries bounties on them because they eat shellfish. They're benthic feeders. And so they'll cruise along the bottom and grab really valuable shellfish. And they were certainly hated in the San Francisco Bay back in the 1800s. You can read about this in Jack London, a California writer. He was an oyster pirate for a while before he became a writer. And he talks about the oyster trade out of Oakland and the pirates and the drinking and the brawling. He talks about pulling up sea stars and cutting them in half to keep them out of the oyster beds that they were harvesting from. Of course, when you cut a sea star in half, you get 2 sea stars because they grow back. And they talk about their war. It talks a little bit about their war on bat rays. The bat rays still live in the bay. The oysters, not so much. They were done in by change. Anyway, that is another strike against the rays. Also, the wings are very popular food. The wings, unlike a lot of sharks, which have this very like urea saturated flesh, because that's how they osmoregulate, by leaving waste products inside their tissues to create tonicity with the water. I've spoken about this before. Bat rays tend not to do that for whatever reason, at least in the rays. And so it's a very popular food item, and you can find them dried, grilled, and they cut them out. I don't know if they still do this in the States, but until fairly recently, they would market them as scallops, which is ironic because they were hated for the scallops that they ate. But they would cut out these little cookie cutter shaped round things. So when you go to a restaurant and you order scallops, if it looks just like a little too round and not really like clam-like, scallop-like, you might be not eating a scallop at all. You might be eating a bat ray or a ray of some sort. Yeah, question? I don't know. I don't know much about it. I think there is a little poison gland at the bottom of it. So I think they can inject a slight amount. You're nodding your head. Yeah, there's a small poison gland with a tube, some kind of tube, so they can inject a little bit. It's probably not the poison gland that'll get you so much as the serrated quality and the bacterial infection that sets in later. Yeah. Oh, yeah. You're from San Diego, right? LA, yeah. Yeah, you're telling them, I'm here, don't sting me. There's no real advantage for them to sting you. It's just a defensive thing. So if you shuffle, it's much easier for them to to shuffle away than to jab you. That's the theory, at least. I don't know how well it works. Apparently it works well enough. So I want to talk about family Mobulidae, which are a very impressive ray. They're the manta rays. And specifically, this is the giant oceanic manta ray, which has a global distribution. It too is endangered according to the IUCN list. It can be up to 4.5 meters long. I don't know what the width on that is, but it's a lot, up to 3,000 kilograms. So these are extraordinary animals, and they're filter feeders, like a lot of ocean giants. They tend to feed low on the food chain. Why? Because in order to support such a large body, you need to be able to get a lot of what's out there. And food and zooplankton is an efficient way to feed because there's so much of it. You're not going through separate trophic levels, losing energy along the way. They have very low fecundity. They give live birth one pup at a time. They have a very late maturity and a long gestation period, and they're very, very long-lived. And so all those things together mean probably not a great fishery, right? It's probably really difficult to harvest sustainably these fish. So even though they have this global distribution, they're very much endangered. That may vary from country to country or region to region, but generally they're in trouble. And so I just want to address this a little bit, some of these ideas. Sharks in general live very long lives. And so I just talked about fecundity and age at maturity and lifespan. Can you, and this paper came out a few years ago. They used bomb carbon dating to compare the originally validated measurements of shark lifespan with bomb carbon. Bomb carbon is a carbon signal left over from the bombs that went off in Japan at World War II. And so and actually there were other bombs as well, but mostly the giant atmospheric signal that we get is detected from the mid-1940s when those bombs were dropped. And so we can see the age at which these fish, you can see the age that these fish are because we can detect it in them. And if there's a detection of that carbon from the bombs, essentially we have a hard date. There's another hard date, a new one that happened with Fukushima in Japan. So a lot of Eastern Pacific fishes, like long-lived fishes, we have a new marker. But this is one way to validate your aging methods for the sharks, which are really hard to tell sometimes because they don't have hard parts like otoliths. Anyway, what they found out, does that make sense? I just kind of stumbled my way through that. So what they found is that like a lot of these animals are very underestimated. Like let's take C. carcarius here. Those are the white sharks. Come on, there it is. So this is the white sharks. So they were underestimating the age of the population almost by half. And that can have really devastating effects on your evaluation of the sustainability of the stock and whether you can actually manage a fishery for it. So can anyone give me an idea why overestimating fecundity would be really damaging in terms of managing a stock? Yeah? That's exactly it. Well said. That's exactly it. So fecundity is easy to understand. How about underestimating age and maturity? How does that affect understanding the population dynamics? Yeah. Right. Exactly. Smaller fish could be harvested that may not have even had a chance to reproduce. And then if those fish have very low fecundity, that means that you're not only letting less fish into the reproductive capacity or the reproductive portion of the population. Once they get there, they have very low productivity. And now I think so that's age and maturity, and then lifespan. So if we underestimate lifespan, how does that affect our understanding of management dynamics for the fishery? Anyone think about that? If you're underestimating lifespan, what does that mean? I think it's a little trickier. Yeah.
Might be underestimating like the amount of offspring that this individual could have in a lifespan, and therefore you're not really understanding
Yeah.
like how the population is getting replaced, or like I don't know, like you're cutting that really short.
So it leads to a lot of error,
Yeah.
right? It's a lot of uncertainty, right? That's part of it. And also, say you're catching like 80-year-old fish that you think are all 40-year fish. And then you think that the representation of the population is in these younger fishes, whereas you have, so like when you look at the population structure of a population, you expect that like there's a whole bunch in fishes, you expect there's going to be a whole lot of babies, less adults, and some of them get killed off, and even less older fish, right? But if you assume that the fishes are younger than they actually are, in things like sharks, you should have a whole lot of baby fish, a whole lot of middle-aged fish. But if they live so long, you should have more older fish than you expect. And if you underestimate that, it means that this whole reservoir, like you would expect for a short-lived fish to have that tapered curve so that you expect less adults, fully mature adults. But in reality, because they're so long-lived and they have such low fecundity, they need a reservoir of really older fish, and they should have a higher representation, a higher proportion of older fish. So in summary, if you have a long life, you should expect there to be more old fish. Humans have a pretty long life. There's a lot of old people out there, right? And there's a lot of young people. We tend to have, well, it's a different life structure, right? But the point is, there should be a lot of old sharks. If you don't think they live very long, you would expect it to be normal to not have as many sharks. And we had that expectation, and it's wrong. And so as a result of this, we're closer and closer to understanding how the fact is that for white sharks and many other sharks, they're way overfished. And possibly for something like a white shark, it's probably not even possible to fish them sustainably at all. They talked about in the Atlantic, they talked about removing hundreds of juveniles, not thousands, not tens of thousands, not, hundreds. That's not very many for an ocean. Hundreds of juveniles per year could have resulted in population level declines in excess of 60%. So sharks are a really tough fishery to manage, and that's why they're so endangered for many reasons. Yes? Usually, yeah. Yeah. Like some kind of biopsy or something, or you can't do a fin clip on it, like with Acanthopterygian spiny rayed fish, you can do a fin clip and get a spine and they'll give you a ballpark, but you have to kill them. So, for a lot of these studies, they're probably not intentionally killing them because they're too rare. Not now, but at one time, probably. And they would take like vertebrae samples, or sometimes, yeah, probably vertebrae are the best for sharks. Okay, onward. All right. Actinopterygii, Sarcopterygii to Actinopterygii. We're going to kind of go through a bit of an evolutionary spurt here. We're finished with most of these ancient fish, and now we're going to look at the transitional fishes to the new modifications that characterize what we call fish most commonly. And so here's where we are. We just finished the jawless fishes, the cartilaginous fishes, and we're going into Sarcopterygians, which include coelacanths, which I've talked a little bit about, the Dipnotetrapodomorpha, or dipnoi, which are the lungfish, and the tetrapods, which you'll have to get somewhere else. That's where we're heading today. And then after we're done with that, we'll talk about the ray fin fishes a little bit. So we're moving down into the Cladistia, which are the Bashirs, the Chondrostii, which are the sturgeon, the Neopterygians, which include gars, and also the bowfins here. And as we move from these ancient fish to these ray fin fish, we'll talk about what happened to them along the way. And I don't know, something happened. It's because I put a video in. Against my best judgment, I put a video in. I have like really strong rules about videos. Like they can't have an Australian accent. They can't have bad music. It's not okay. They Go away. Stop it. This video is really good though, so if I can get it to play, we'll watch it. All right, so the coelacanths is our first group. Coelacanthiforms, there's one family and two species. And those two species, the first species, the Latimeria chelumni, were found off the coast of, oh, damn it. God, sorry, I shouldn't swear. Being recorded. Latimeria chelumni is found off Africa, and Latimeria menadoensis is found in Indonesia. We've talked a little bit about them. They're like dinosaurs, right? Everyone says it's like a Tyrannosaurus Rex, and that's the exciting thing about them. Even though they're not really the missing link to the tetrapods, they're really cool because they thought they were the missing link, so people got really fired up about them. And you know, they had only known them from fossils from the Jurassic period. I mean, they're really, really ancient. And so some of the cool features about them is that they have these things called cosmoid scales, which are only found in extinct species. And you can find them on these living fossils. All the fins are jointed. They have these homologous structures to the humerus in your arm and the femur in your leg. They've got those structures. So we have a common ancestor somewhere way back when, from which they and us evolved. And they also have like this fatty vestigial lung. So they have lungs, but they didn't use it for breathing. The fat in them helped to regulate their buoyancy in the water column. And so they have these two remaining species that I mentioned before. They're both extremely rare because they live like 80 to 100 years. They carry their oviviparous, and so they carry their young between 14 months and 5 years. We don't really know. It's amazing. And then they give birth to 5 to 26 live young. They're very deep water fishes. They have a very slow metabolism. They probably survived all these years because they live very, very deep in the water. They don't need a lot of food to get by. And things just haven't changed that much where they are. And so they persist because they found a weird niche. And the weird niche has persisted for millions and millions of years. Oh, I mean, did I say that? They don't reach sexual maturity until they're 20 years old. Yeah, it's just they're kind of an amazing animal. And then on top of it, I was reading about them, and they're blue colored. So all the ones that we see that are pickled, all the pictures, they look so drab and ugly. Turns out that when they're fresh and new and just brought up, they're this intense blue color, 6 feet long, as tall as a person. So I'm going to take a little bit of time then to play this video in the last little bit of the class here. Of course, I'm not getting as far as I wanted to get, but actually I was really moved by this video. It's like I think it's a really good one to watch for a lot of reasons. So let's try, because I think it captures all right, be patient with me.
It was the 21st of December 1958 when Himmelkreuz, the captain of early advancing trawler Marie, made their last haul in about 70 meters of water.
Sorry, unbelievable. I won't touch it.
It was the 21st of December 1958 when Himmelkreuz, the captain of early advancing trawler Marie, made their last haul in about 70 meters of water off the mouth of the Chilena River. Fish with strange fins and an excellent time of the steak. The surrounding largely with the atom was curated after the Smithsonian Museum. Person was in the habit of saying anything unusual about the statue saw there. Next morning, seemed to have a pile of shark stories and other specimens. She found the blue fish. After having brought the fish back to the museum, she tried to identify it, but without success. So she sent the picture of the statue of the fish to Professor J.R.B. Smith at the University in Brainstorm. It was called a genomic code of about 40 colors. Described in heavy scales, almost almond-like. The figures resembled wings, and scales right up to 40 millimeters. The spinal dorsal has tiny white spines down each finger. It did not take them long to decide that it was a living fossil, a coelacanth. Coelacanths were thought to have been extinct for 70 million years. Finding a live coelacanth was a little like finding a live dinosaur walking around the Peru. The fish met a area to Limerick in her honor at the river where it was found. As these were the museum did not have a freezer or preserving tank large enough for fish, its internal organs were discarded. A second, intact specimen was needed. By the late 1940s, no more coelacanths had been found. Smith supposed it was because they lived in some more remote part of the western Indian Ocean. So he had a poster designed and distributed in various islands in the Mozambique Channel, offering the reward of £100 for a coelacanth. In 1952, able J.R.B. Smith evacuated the Comoro Islands to tell them coelacanth. The fish would not be properly preserved. Smith enlisted the help of Dr. George Milan, then Prime Minister. Milan approved the use of an Air Force airplane to take Smith to the Comores to fetch the fish. Since 1952, more than 180 coelacanths have been caught in the Comores, but not again in South Africa. Then, on the 28th of October 2000, 3 amateur divers, Peter Venter, Peter Tim, and Etienne Leroux, were exploring Joseph Canyons of Warren Bay at a depth of 104 meters. Nearing the end of their dive, Venter saw a large eye reflecting the beam of his underwater torch. Underneath an overhang, he saw a large blue fish. He was convinced it was a coelacanth. He summoned Tim, and they saw 2 more. A month later, they planned another dive, this time taking a video camera. They found 3 more coelacanths at 108 meters and filmed them. We have at last discovered coelacanths living in South African waters. Just as the very first discovery of the coelacanth sparked an explosion of research and scientific endeavor, the first living coelacanth to be found in South African waters has fueled the creation of the multi-stakeholder, multidisciplinary South African coelacanth conservation and genome resource program. The program was launched in April 2002 at Sagara Bay by the Department of Arts, Culture, Science and Technology through the National Research Foundation. Yes, Dr. William Gavani explains why the South African government got involved in the project.
Well, because it's one of the major issues in this field today. And this offers us a connection with the past, going back to the age of the some very few countries have such an opportunity. We will see this as a reflection.
So, you know, I wanted to go to the end of that. Thanks for hanging in there. Got 1 minute left. And I just want to say the reason that I put this is because I kind of found it just like it encapsulates the sort of excitement of discovery that often gets lost, I think, in science and dry lectures like mine. But also the fact that they found this fish and then it became an emblem for the country as a source of national pride and conservation. And today they're using it as part of outreach programs to kids to value conservation, not just for the coelacanth, but also for other aspects of African culture and African wildlife. And I thought, God, that's so, it's so inspiring because that's where we should be as, you know, growing conservationists ourselves, that we should be using our model organism, whatever it is you're into, being able to use it as some kind of a hook to get people in to value the things you value and to like create a movement around it. And that's much more exciting than like, you know, Shark Week, for example, you know, at least to me. Thank you. Have a good afternoon.